currently is a pest in Spain, Hawaii, and in Southeast Asia. So my group and I are interested in finding ways to eradicate this snail, to stop its propagation, but for that we thought that first we need to know its physiology, so we decided to study the digestive system of this snail with proteomics approaches. So for that we took samples of different tissues, of digestive tissues as salivary gland, digestive gland and intestines, and took digestive contents, crop and style sac. Our workflow was we did protein extraction, and then filtrate sample preparation, followed by the MS analysis. So as you all know, working with non-model animals is really hard in this field because of the lack of database. So for that we had to build our own database, and we looked for all the proteins that are in public sources for mollusca, and also we found the transcript of Pomacea in an open source, and we built our database. So the results that we got were proteins matching with uniprot accessions of the mollusca proteins or IDs of the transcript. But the difficult part was the ones that matched with the transcript were not annotated, so we needed to do a blast alignment. So as you see in this picture, this is the results that we got for the digestive gland. And as you can see, few of them were matching with the mollusca database of proteins, and a lot of them matched with the transcript. So that means that we needed to do a lot of blasts. This table shows all the numbers that I got for all the tissues and the contents, and as you see, these are the total numbers of IDs, and they are really high, and the total number of matchings with the transcript are really high too. So we started thinking that maybe something was going on with the database, and we realized that some of them matched with the same accession when we did the blast. So as you see in this example, we realized that some transcripts match with the same accession number, and there was an overlapping that you see the green part on this. So we needed to do an extra curation of the data. Then we did the ontology of the digestive gland, and as you see, the proteins of metabolic process were overrepresented, and this is in accordance with the function of the digestive gland, and also because Pomacea needs high energy demands for inhalation. So the next that we did is finding all the proteins that have a digestive function, that participate in digestions, and we classify them due to the target substrates. And we realized that the digestion of carbohydrates, it's really, they have a lot of IDs that participates in this digestion than protein digestion. And also the contents, even though that they don't have as much proteins as the tissues, they have high amounts of proteins participating in digestion, especially in the carbohydrates. So then I classified the proteins participating in protein digestion, and due to the class of proteins. And I found out that endopeptidases are really high in the digestive tract, and serine peptidases, that are the first one here, are keep constant during the digestive tract, and also are the main peptidases found in the contents. And in the tissues, the main peptidases found are exopeptidases, and these ones are aminopeptidases. So my conclusions and the corrections are that the golden apple snail is a non-model organism and the protein database are incomplete. To improve our identification rates, we build a custom database using the data available in open sources, but this yields to 10 times more IDs than the ones that were really there. The transcripts IDs are not annotated, so we needed to do the blast. However, the transcripts are fragments of proteins, and as such, we know that inflation of the total number of proteins due to an overlapping of the sequences. An extra curation of the data was required to remove all this redundancy. And when we analyzed the data, we found a broad diversity of peptidases through the digestive tract, where serine peptidases were the main enzymes in the digestive contents. Also, the carbohydrate digestion was apparent the main digestion. So we hope that this study could lead to a better application of molluscicides and understanding of the physiology of this snail. So thank you very much for listening, and thank you to my supervisor, Michelle Colgrave. Thanks.